Okay, hello everyone, welcome back. So this is the start, technically, of a new topic, the old genes, genetics, and history. A little bit, so you're getting into evolution and speciation and so on and so forth. But it linked perfectly into the previous GCSE biology genetics video, as it's basically how we got to know all of the amazing things about genetics that you've learned over the last few videos. Okay, so what we're looking at today is essentially history, essentially a timeline of how we've got to where we are, because people have always considered, how wondered and worked on how life works, how things get passed along, and where we are, where we come from, and our place in the world. So I've subtitled this "The Search for the Secret of Life," and as you'll see, and amazing facial hair on some of these people. Okay, so what do you have to know? Well, you've got to know basically a timeline that starts in the early 19th century, mid 19th century, with a monk in Austria, and ends up with glow in the dark rats and bacteria produced insulin. Okay, so that's basically what we're talking about here. We're going to go through the steps in the middle, and you can see the lines on the spec there. So let's start off then. Right now, obviously, this story goes way further back than the 1800s. Okay, well, so we start in the 1800s, but of course this goes, the search for sort of answers and heredity and family and where we come from and all that kind of stuff via various both scientific and non-scientific methods goes back millennia. Okay, so as a recommendation for you, as a writer that I really rate, a guy called Steve Jones, who is a prolific science writer and science communicator. For example, I mean, I read this book when I was applying for my degree as preparation for my interview, and I actually saw him only the year before last at a conference in Birmingham. Right, so that's his career has spanned that length of time, and he postulates in this book, if you go into any sort of historical book and replace the word blood with the word genes, or take, don't expect to look for the word genes, look for the word blood, you'll actually see that people are talking about inheritance, heredity, genetics, and so on and so forth, way before the start of our story. So I recommend his books for those of you who are super keen on understanding a little bit more about the sort of deeper history of this. So let's start straight then, jump straight into the 1800s. So the mid 19th century we are, and we're going back to this guy. Okay, now this guy was mentioned in the inheritance video, Gregor Mendel. When you think genetics and you think inheritance, his name is the first one that jumps to mind. Now he was an Austrian monk, mid 1800s. He was working, monking around. His name is Gregor Mendel, and did experiments with peas, as we said in that previous video. Now, mid 1800s is the 19th century. I hope you're all okay with that. The idea that 1800s, 19th century, same thing, because 0 to 99 was the first century, and then 100 to 199 was the second one. We all happy with that? I hope so. Anyway, right. So moving on, he didn't look at things that you've looked at in the previous topic, like eye colour, hair colour, blue and brown eyes, all that kind of stuff. He looked at smooth and wrinkly peas, or pea plants that were tall or dwarf. Okay, now that's a very slow process. Pea plants, you can get 2, maybe 3 generations out of a pea plant per year. They don't grow very well over winter, of course, and of course they're not exactly very applicable to us, to humans, so for medical purposes, which is why we've previously said nowadays we work on things like Drosophila flies, nematode worms, things like that, to do our sort of genetics and inheritance experiments. So what did he do? Well, I've shown you this diagram before. This is our famous 3-to-1 ratio thing. So what he did was he crossed a load of plants that were what he called pure breeding tall. So basically you bred them for loads of generations and you just got tall plants, and then colonies of pure breeding dwarf plants. We now know that we would apply words like homozygous dominant and homozygous recessive to these ones, because he should have done that, learnt that in the previous videos. And he crossed those, and he found that he never got any plants that were halfway in between. He got all tall plants. Now you should obviously know now that that means they have a dominant gene and a recessive gene. They are heterozygotes. But he was surprised at the time not to get medium height plants, and he thought, hang on a minute, what's going on here? So then he crossed those offspring, back crossed them, and ended up with these ratios quite reliably, right? Got small or tall in a 1-to-3 ratio. 1 small, 3 tall in a reliable, predictable ratio. Now, people have replicated his experiments since, and either he was very, very, very lucky to get experiments that were results that were as good as he got, or he faked it a little bit, right? He sort of massaged his figures and made them look a little bit better than they are, which is the suspicion, right? Naughty, naughty Gregor Mendel, naughty, right? So he got this ratio, and he basically didn't know anything like genes or DNA. Of course, he called them units of inheritance. So he postulated there must be something in these P's that gets passed on that is dominant or recessive over a different version. Okay, so he came up with this idea of dominance, recessiveness, these ratios, and this unit of inheritance. But unfortunately, nobody really cared. So he wrote a load of letters to various botanical and scientific institutions, wrote a book on this, but nobody really cared. They were like, yeah, great, whatever, right? Because he wasn't a scientist, he wasn't working at a university, he wasn't a respected name, he was just some monk messing with his peas. So nobody really paid attention. They were like, great, you found some short peas. And that's often the way it goes with science, that important discoveries aren't realised for how vital they are until much after the time of the discovery. So let's have a look at the Punnett squares, because this is something you reasonably could be asked to do on a GCSE. Okay, so let's look at his so-called pure breeding strains that he called them, which we now know would be homozygous dominant, homozygous recessive. Cross those over, look, right? You, as you know from the previous lessons, right? Then you put the gametes here that they make. Now, obviously, it's not sperm and egg; it's pollen and egg in a plant, but it's the same principle. So then, if this gamete meets with this gamete meets with that gamete, then you get all of them as heterozygotes. Okay. Now, what you can do at this point, pause the video and answer this question. Now work out what genotypes and phenotypes the peas in the second generation would have. Okay, so that's quite straightforward to do. I'll give you a moment to do that. Hopefully you paused there if you wanted to work out the second generation, because it's something you can reasonably be asked to do on a GCSE. Well, of course, all of the parents will be big G, little g, so when you make the gametes, they'll look like this. So offspring 2, these are a cross between tall green peas, big G, little g, right? And you get this, of course, the famous cross. Now we can replace that with anything, brown eyes and blue eyes, any other dominant or recessive cystic fibrosis example from your genetic diseases one, and that would be absolutely fine. Okay, so in terms of genotypes, you've got one big G, one homozygous dominant, two heterozygotes, and one homozygous recessive. But in terms of phenotypes, what they look like, you've got 3 green peas and a yellow pea. Okay, easy peasy. However, right, Gregor Mendel overlapped mid 19th century, right, mid 1800s. That is, of course, the time of the Grand Maestro, and I imagine you should know who I'm talking about. Okay, that's kind of a contemporary picture of him there, right, of when he would have been working with this guy. But if I take that picture away, maybe you'll have a bit of a better idea of who I'm actually talking about there, or one of his more famous older pictures. I am, of course, talking about Charles Darwin, right, that he was around at the same time. So Darwin had got back from his voyage. We're not going to go into Darwin's voyage, that's a whole separate lesson. Darwin and Wallace require a whole separate lesson for this. The evidence is, from looking at the letters and various things, that Mendel knew very well who Charles Darwin was, because he was a respected name, a member of the landed gentry in the UK, and a well-known man of letters and writings. And this book he produced, The Origin of Species, Mendel had it, and there are versions of it where Mendel has actually annotated and said, "Oh, I agree with this. This is like my peas," and bits where he's gone, "No, I don't think that's right." So if you read The Origin of Species, the bits on what is now genetics are quite weak. Okay, so the evidence is that Mendel knew who Darwin was, but Darwin wasn't really aware until quite late on who Mendel was, when someone went, "Hey, your book about the evolution, maybe you should read this Austrian monk, what he's been saying." But I don't believe they ever met. Okay, so we'll leave Darwin, right? But obviously probably know a lot about him already, and we'll come back to him in another lesson, and we'll skip on. Okay, we'll move on. So second stage of this, so you've got your monk and his peas and his units of inheritance and so on and so forth. Next thing to happen was a little bit later, 30 years later, was the discovery of chromosomes. Okay, and this is a dude called Walter Fleming, and I hope you're seeing now why I've called this and the search for amazing facial hair. Right, we're now getting into peak facial hair time of this lesson, So Walter Fleming was a German biologist, and he was the first person to see chromosomes down a microscope. Now, around this era, there were various people looking at cell biology and working with cell stains, and there was some awful stuff done with old bandages from soldiers in the Crimean War, looking at infected wounds and looking at the cells in there, which turn out now we know to be white blood cells, and seeing chromosomes in there. But Walter Fleming was the first person to actually see down a microscope in 1879 these thread-like structures, which he called chromosomes. Okay, and he looked at them, he watched them in many different cells, and he first described the stages of mitosis as how cells divide. Again, something else you should now know and take for granted as your GCSE biology knowledge. Okay, and he put out this book. I'm going to mess up the German here. Zell Substanz, Kern und Zellfügung, where it's cell substance, nucleus, and cell division in 1882. Right. So he put that out, and obviously everyone thought this was a great book. Look at what amazing things he's done in microscope work, but no one actually really properly made the link in common scientific between him and Mendel. And hey, do you think maybe these chromosome things could be something to do with these units of inheritance that this Austrian monk was going on about 30 years ago? No one actually put the two together for about another 20 years. So after Fleming, right? So anyway, this is some of his videos, some of his images here. So on one side here, you've got Walter Fleming working in the 1800s, so 1879, drawing the stages of mitosis from his ancient microscope. This, for comparison, is what a Year 12 student can do in biology now. Okay, so this is with smartphone cameras, proper modern microscopes in a lab, best possible stains, well-defined protocol, all that kind of stuff. So that's one of my Year 12 students' photograph of cells going through mitosis, and this is what Walter Fleming managed to produce, over 100 years before. So incredible work given the technology available at the time. So clear spindle fibers here, clear chromosomes. So just an amazing piece of work. Okay, and I say this wasn't recognised at the time. I was 20 years, but actually there was one guy who did go, hang on a minute, this is important, and that led us into stage 3 of this story, where we're going into the 20th century, the early 1900s, the dawn of the so-called gene age. And this is where we've also got some fantastic facial hair. Okay, so this guy Wilhelm Roux in 1883, he speculated, so 1883, remember that's 1 year after Fleming's book came out, he speculated that chromosomes might be the carriers of inheritance. He was like, hang on a minute, this inheritance malarkey could be to do with these chromosomes. But that didn't really get any traction for about another 20 years. So now we're jumping to 1905 to 1909, where various scientists are working on this topic, and that's kind of how science works. A topic becomes fashion, it becomes a hot topic, you get lots of research money and lots of researchers working on it. So a Danish botanist called Wilhelm Johansen coined the word gene to describe these fundamental physical and functional units of heredity. So he still didn't know anything about DNA, right? But he thought, hang on a minute, maybe these chromosomes contain units that match with what this monk guy was saying, right? And these units, I'm going to call them genes, okay? And William Bateson, around the same time, coined the word genetic, right? Genetic, to describe patterns of movement of these units of inheritance. So now we've got chromosomes, we've got the words genes and genetics, and we've got the idea of dominance and recessiveness and things moving down generations in patterns, okay? And basically heredity. So now the stage is set, right, for jump 50 years in the future, of course, because scientific research in the early half of the 20th century was somewhat hampered, if it didn't make a bomb or a weapon, right, by the World War I and World War II. So now we jump forwards to the double helix. Now, looking at all these fantastic beards and moustaches and sideburns, you might rightly ask the question, where are all the women in this story? Why are we just talking about a bunch of bearded men, right? And the answer is, well, of course, patriarchy and sexism through history, but here is where we do get the story of the first majorly significant woman contributing to this story, and it is a bit of a controversial story. So we're now in 1953, okay, the work leading up to 1953. So 1953, these two guys at the top, Watson and Crick, come running into the Eagle pub in Cambridge. It's a lovely old pub. If you ever get to go and visit it, there's a plaque on the wall commemorating this moment, claiming they've discovered the secret of life, the absolute secret of life. They discovered that most famous of images, the double helix. Okay, they basically characterized and built an accurate model of the spiraling twin strands of DNA with all their bases lined up in the middle, as you've learnt about over the last few videos. Okay, and they came running in there, and this was a major discovery, and they got a Nobel Prize for it, as well as this guy, Maurice Wilkins. Now these guys here, right, Wilkins and Franklin, were working down in London, right, and these guys were working in Cambridge. And the story goes that these guys here were reputedly very lazy in their approach. They were geniuses, they were very, very smart guys, but they didn't like to do a lot of experimental work. They liked to take other people's data, put it together, and come up with inferences and insights that no one else had come up with before. And they were very, very, very good at it, right? As most people who get into, let alone work at Cambridge, are. Okay, these two here were doing the work. Particularly, Rosalind Franklin was doing work with X-rays, a thing called X-ray crystallography, and that generates pictures like this. And it's this picture here where all the controversy is. Okay, so she's working and working and working away, right, to generate these pictures, like taking bits of DNA and putting them in little crystals in a paint can and firing X-rays at them, and basically looking at the pattern of the X-rays around the inside, pictures like that, and trying to work out what pictures like that mean. And she'd got these beautiful X-ray images of the structure of DNA, but she couldn't put that together, she couldn't work that out. And then Maurice Wilkins, unbeknown to her, got hold of her images and passed it to Watson and Crick. And it was the last piece of the puzzle that let them work out what the structure of DNA was. Okay, so her unpublished work was passed without her knowledge to Watson and Crick, who then worked out what the structure of DNA was and received a Nobel Prize. Now, the rules about Nobel Prizes are they can only be shared between 3 people, and you can't get them if you're dead. Now, unfortunately, all this work, right, with Rosalind Franklin did with X-rays, right? X-rays, obviously, in high quantities over long periods of time are quite dangerous, and she died in 1958 before ever receiving credit for her absolutely pivotal contribution to the discovery of the double helix. And it is a story of 1950s sexism, entrenched patriarchy that has come down since then as to whether Rosalind Franklin was robbed or not. And it's not a simple answer, right? The answer is in possibly yes, possibly no. There were certain things that she hadn't worked out that Watson and Crick did, but the way that that data got passed to them was certainly not normal scientific process. So there's tons of books, documentaries on this story here, and you can look into the personalities and the people behind this story. But the long and short of it is 1953, we had the double helix. So in that time, between 1850 something and 1950 something, that 100 years, we've come from not knowing anything about DNA, genes at all whatsoever, to understanding inheritance, to knowing what a gene is, to knowing what chromosomes are, to knowing how heredity works, how chromosomes get passed down the generation, and understanding the double helix. Okay? And that is the series of events that is on your syllabus. Obviously, it didn't stop there. In fact, this was only the beginning. You are living through an amazing time to be a biologist, possibly one of the most powerful times to be a biologist in the 4 billion years of life that has existed. Okay, so to summarise that story, the discovery of the double helix, as you can see here, look, mid-20th century, 2 rival teams of scientists are trying to solve the riddle of inheritance. In London, Wilkins and Franklin. In Cambridge, Watson and Crick. Crick and Watson working on 3D models. Crick and Watson managed and came up with the double helix structure, but they only managed this by using Franklin's X-ray results released by Wilkins without her permission. Watson, Crick and Wilkins all received Nobel Prize. Franklin died before these prizes were awarded. Okay, so do go and look into that, come up to your own conclusion as to whether she was a victim of massive 1950s sexism. Okay, so just as a coda, an end to this lesson, right, since then we have basically had a genetic revolution. These discoveries have caused an explosion of amazing discoveries and amazing things that people can do. So we've had the Human Genome Project in the early 2000s. Okay, you can now go to a GP and be genetically tested and counselled as to what your genes are going to be like in the next generation, what chances you have of developing diseases, what chances your children have of developing diseases, as you will know from previous lessons that you will have gone through, right? Genetic engineering, of course, is massive. Right? Since 1994, I believe it is, if you're diabetic, you now receive your insulin from a genetically modified bacteria that has a human gene in it and manufactures that insulin for you. Okay? There's a field called synthetic biology, which allows us to stitch together genes in whatever order we so choose. Okay? Which means that we've now got this thing called spider goat wandering around the planet, a highly controlled farm, which is a goat that in its milk can produce the protein for spider silk. Threads can be spun, and that can be used in medical applications. The spider silk's incredibly strong. We've got glow-in-the-dark mice. In fact, we can knock up glow-in-the-dark bacteria in a school lab in an afternoon. It's not a problem with genes from a jellyfish imported into bacteria. You've also got the possibility of gene therapy, of using augmented or working genes to help cure anything from cystic fibrosis through to predispositions to cancer. And you've got gene editing down to the base level, this tool called CRISPR-Cas9, which if you're interested in is the big thing. Go and have a look at it, right? Go and have a look at what it is, where you can literally, to the base, edit genes to correct issues or augment or enhance different genes, okay? All that starting with a monk in his greenhouse mucking about with peas. So it is an amazing time to be a biologist, right? This is the hot science, I would argue, that exists now, right? So obviously there's loads of different stuff in that video that you could go off on a big tangent about, and I'll put some links in the description to various places that you can find more information, okay? We'll leave it there, and I'll see you next time. Thanks very much.